Hello everyone, and welcome to another tutorial from iOS E1A. This tutorial is about understanding how the cell reacts to change in membrane fluidity. In this question, the bacteria will have some responses when they are transferred from 37 degrees Celsius to 25 degrees Celsius, so they are moved to a colder environment. We will go over a few responses, and we need to explain why each of the following responses is or is not likely to be observed. The first one, initial decrease in membrane fluidity. So at colder temperature, does the membrane fluidity decrease? Yes, this response is likely to be observed. Fluidity is temperature dependent. At lower temperature, the molecules move more slowly and pack together tighter, making the membrane less fluid. How about this response? Gradual replacement of shorter fatty acids by longer fatty acids in the membrane phospholipids. For example, we'll be making more stearic acid to replace the shorter palmitic acid. No, this response is not likely to be observed. Remember that at lower temperature, the plasma membrane is less fluid, so the cell is now responding to increase its membrane fluidity. To do so, the cell would need to reduce the strength of the van der Waals forces between the hydrocarbon tails. And to reduce van der Waals forces, you will need to reduce the size. So the bacteria should be replacing the longer hydrocarbon tails with shorter hydrocarbon tails, and not the other way around. How about this? Gradual replacement of stearate, which is 18 carbon atoms and saturated, by oleate, which is also 18 carbon atoms, but with one double bond. Yes, this response is likely to be observed. Again, the cell is trying to increase its membrane fluidity by reducing the strength of the van der Waals forces between the hydrocarbon tails. Another way to reduce van der Waals forces is increasing unsaturation, making more double bonds. Saturated lipids can stack up neatly, decreasing fluidity, but unsaturated lipids form kinks, disrupting the fluidity of the membrane. How about this response? Enhanced rate of synthesis of unsaturated fatty acids with double bonds. Yes, this response is likely to be observed. Similar to the previous response, the cell is trying to increase the membrane fluidity by reducing van der Waals forces, which could be achieved by synthesizing unsaturated fatty acids. How about this? Loss of integral proteins from the membrane. No, this response is not likely to be observed. You would not expect temperature change to affect protein content. How would the presence of cholesterol in the membranes of the bacteria affect the changes in fluidity that occur as a result of the initial temperature change? So cholesterol is a bidirectional regulator of membrane fluidity. In high temperature, cholesterol reduces fluidity, and in low temperature, cholesterol increases fluidity. In high temperature, cholesterol reduces fluidity, stabilizes membrane by holding phospholipids together, raises its melting point, and restrains movement of phospholipids. In low temperature, cholesterol increases fluidity, intercalates between phospholipids, and prevents tight packing. So the decrease in membrane fluidity that occurs as a result of the drop in temperature would be less extreme if the membranes contained cholesterol. That concludes this tutorial. Thank you for listening, and good luck.